Hope everybody had a chance to grab a snack or take care of business during the break. It was so great to have such an interesting keynote, and we're going to continue on with this theme of research on pollinators with our morning session. And so you guys have already heard me talk a lot today, so I'm not going to do a whole lot of talking or introductions, other than to just say that we're really lucky to have representatives from the Minnesota Bee Atlas. I'm hoping I've got the right project there. We're going to hear from 3 different people about multiple aspects of projects going on in Minnesota. And maybe you'll hear a little bit about some projects that were related to things you may be familiar with here in Colorado. So without further ado, I'm going to start you off with Britt Forsberg, and I'll let each of the speakers kind of tag off and introduce their collaborators, and I'll let her take it away right now. Thanks so much, Britt.
All right. Thumbs up that our presentation's up on screen. That's all going for everybody. All right. Hearing no argument that that's not the case, I'll get us started. So yes, I'm Britt Forsberg. You'll hear from my colleagues in a little bit, but I will introduce you to the Minnesota Bee Atlas. So this project was a 5-year grant that ran from 2015 to 2020. We had 4 staff positions, 1 full-time. We had 2 part-time bee ecologists, and then a lab tech who worked with us, as well as we were able to hire an independent web designer. So the goal of this project was to work with volunteers across the state to collect baseline data on native bee distribution and diversity. We wanted to take all this information and make it available in a public database. There's a ton of bee research happening in Minnesota, but it's hard to find all those individual pieces. And using volunteers, as you'll hear, really helped us get at some unique data points. So the 3 different protocol areas, I'll talk the most about using iNaturalist as a photo-based collection tool. But I was the program coordinator. I had the 1 full-time position on it and did the most interfacing with volunteers. So that meant recruiting volunteers, finding people who had appropriate skills, interest, lived in a portion of the state where we wanted more data. Minnesota, people love bees, but it wouldn't help us if we had everybody in our major metro area as our only volunteers. Once we had volunteers on board, I managed most of the training, going out primarily in person, but at the tail end, of course, with COVID, doing some virtual events, supporting volunteers as they went through the season. So keeping them up to date with any changes, anything they might need to know about weather, where we were in the season, additional information that helped them do their work better, how their information fit in with what we were finding broadly across the state. So sending out email newsletters, keeping social media going, and then answering any individual volunteer questions. So but again, that was all over for all protocol areas, but I'll talk just about iNaturalist, and then you'll hear from my colleagues Colleen and Elaine about our other project areas. So if you are not familiar with iNaturalist, it's an incredibly powerful tool, both a mobile app and website that relies on photograph observations. So once any user has an account, they can take a picture of any natural sighting. So proof of an organism. This could be a picture of the organism themselves, could be a track, a nest, maybe broken eggs. These could be alive, could be dead. I have several roadkill observations. This is how I get my best mammal pictures, because they can't move and get blurry on me. And then, so it goes into this incredible database. And one of the unique parts about this compared to some of the other online collection tools like this is that the original user does not have to be able to identify the organism themselves. But that photo is there, or in the case of things like birds, you can have audio recordings. But so the community as a whole can help with that identification. The first step is that iNaturalist has created an artificial intelligence called the identitron. So they'll compare the picture that comes up with other pictures in their database and suggest an observation. Sometimes this is at the species level, sometimes the genus, sometimes the family, sometimes it will say, we really can't tell. So there's a suggestion from the program, but then that picture is out there for anybody to find. And so they can continue refining that observation or the identification to make it as accurate as possible. Sometimes this happens by sort of interested members of the public, but in the bee world, there are a handful of particularly experienced people on there. So John Asher goes through and adds a lot of identifications. I've seen Jason Gibbs on there. So they really are people who know what they're doing, and it's not just sort of a guess on the part of the volunteer. So iNaturalist is constantly changing. It's real time. As soon as you click submit, that observation is up there. And so there's no lag. Because of this, we were unable to add it to our final database. But again, it's already a publicly accessible website. And so anybody can search for the parameters they want. They can download the data that they're interested in. When we started the project, the way that iNaturalist handled specific projects was that users had to opt in. They had to choose to send their pictures into that particular project. So that was a lot more work to go find people, to tell them how to use iNaturalist, to make sure that they knew to check the box, to join the project, to send theirs to us. Somewhere along the project, iNaturalist changed, and so they essentially just used filters, which meant I could set the project parameters saying that I wanted pictures that were in Minnesota. The dates weren't important to me, but I needed them to be in one of the bee families. And then it automatically funneled all of those into one place. And as you can see, as of last week, there are over 27,000 observations that came in this way. That really simplified things from a volunteer standpoint because we didn't need to tell everybody about the project, right? People who are just out enjoying nature, taking pictures of bees as they saw them, were automatically joined. And it really increased the number of observations that we had. All, of course, is not perfect with this. These are the top 10 bee species that we found in iNaturalist. And if you look, you notice 9 of them are bumblebees, and 1 of them is a honeybee, not native to Minnesota. So this works particularly well for bees like bumblebees that are large. People notice them, even if you're not out particularly looking for bees, you hear them, you see them. They're identifiable by photo a lot of the time. There, of course, are some exceptions, but these are things where even if the user wasn't able to identify it, someone else was able to come and say, oh, sure, that is this. There are some problems with photo quality. If any of you have tried to take pictures of bees, they don't always pose for you. They don't wait for you to get a picture of the side, of the head, of the different parts that you want to do ID. Or if you get a nice bee, sometimes the wind moves that flower. So there really was some variable photo quality. Some of that could maybe be addressed in the future by providing more resources or more practice with photography. And some of that, I think we're just going to have to say, it's a hard thing and it's not always going to work. There are also many species for which photo identification from a live specimen is incredibly difficult, right? You're probably familiar with some types of bees that even when you have a specimen in hand, ID can be challenging. And so expecting that an amateur scientist member of the public with their cell phone would get a good enough quality photo to identify it is probably not realistic. Another thing that probably influenced the bees that showed up in our top 10 most observed is the establishment of the rusty patch bumblebee on the endangered species list. It is the state bee of Minnesota, and so people are very aware of it. They're out there looking. They're hopeful that every bumblebee they see is a rusty patch bumblebee. And so that increased public consciousness probably increased the number of bumblebees that we saw on iNaturalist. So that all being said, iNaturalist was still a really helpful tool as part of the overall project. So there were a couple of rare species that showed up. The
Hey,
first photo.
Hi, this is Adrian. I was just wondering
Yes.
if you wouldn't mind, would you mind turning your camera off? So for the presenters here that are remote, turns out that if we leave our cameras on, then it makes the slides really small and difficult for people to see. Perfect. Now, everybody, if you can just, anyone else who's in the session presenting, if you can just turn your camera off, we'll be able to see just the slides and it won't be so small. Thanks so much, guys. That looks great.
Sure thing. All right, so we did find several rare species. So 2 bumblebees, one that had not been documented in Minnesota previously, and one that we had a very old specimen in the University of Minnesota insect collection for, but hadn't been seeing. And it turns out they were there. We just weren't looking for them. We also found Megachile sculpturalis, which is an introduced species, so important to know when these things are coming in. It was an incredible volunteer engagement tool. In the lower right-hand corner is a stat from our highest observer. So he had almost 2,100 observations. He is a retired man who goes out with his wife and looks for bees just because he thinks it's fun. He doesn't have a fancy camera. It's a small point and shoot digital camera. Very sweet, gives all the credit to his wife because she's the one who sees the bees. He only takes pictures of them and puts them up. But he travels across the state to look for new places. He will contact the managers, the land managers at a site, and let them know what he found. He'll share his list from iNaturalist. He'll point out what seems particularly interesting or unusual. And so he's really highlighting this to, again, land managers who might not be as familiar. Next up is sort of the dashboard for another volunteer who took our level 2 bumblebee identification course. And he now goes out, he looks at all the bumblebee photos in Minnesota that come up and adds identifications to as many as he can. So he has over 18,000 identifications in iNaturalist. And he's really good at this. These are accurate when I look at them. He sometimes notices small differences that I miss. And then the last image in the upper right here is just looking at the number of our observations in the bee atlas that were considered research grade. So to be research grade, a photo has at least 2 out of 3 observers who have agreed on the genus and species. It has a reliable location. And so the algorithm has determined that that's a pretty reliable ID. So almost 2/3 of our photos were able to be identified to species and agreed with multiple users. iNaturalist will automatically send those observations to GBIF, and Bison can track them too. You can find them on Discover Life as well. So other entities have agreed that this is a worthwhile place to get data from. So in fact, we found all of this so useful. We've continued the Bee Atlas project in iNaturalist, even though we're no longer running that grant. We started a new project looking at the plants that leafcutter bees are using to build their nests. So if you go out and see a plant that has a lot of circles cut out of it where the bees were collecting those plants, and realizing these projects can happen even without dedicated staff time, right? They're out there and users can still add to them. So with that, I will pass to Colleen.
Hello, this is Colleen. Let's see if everybody can hear me. I am going to talk about our stem nesting bee portion of the Bee Atlas project. So my job on the project was to help with the rearing and identification and the methods of this part of the project. The goals were to get an idea of what species were out there and look at distributions of where they were over the state, and also to get some understanding of the timing of when these bees were nesting and when they were active. And I have to say, right up front, we got a lot of inspiration, really good conversation with the folks from the Bees Needs project out of the University of Colorado Boulder when we were starting up this project and getting the methods going. It was really, really beneficial and really helpful to talk to those guys. So thank you very much. Next slide. Britt's going to run the slides for me here. So how did it work? From the volunteer side of things, they had a few responsibilities. It was like a summer-long commitment. So their first activity was to receive and hang a block. Sometimes they would pick it up, and sometimes we would actually mail them around the state. And the blocks had 30 different holes in them of different sizes. We were aiming for semi-natural habitat, so we didn't want very urban places with a lot of people. We wanted to sort of represent the natural habitats of Minnesota. Volunteers were asked to watch a training video on how to monitor their blocks and then learn the different types of plugs that bees and wasps use to plug the outside of their nest. And this is used because that's what's visible at the end of the hole when the bee is done nesting. And there's different materials that are used by different groups of pollinators. And I do say bees and wasps because about half of what we got are wasps. And these are solitary nesting wasps that have a similar life cycle to the bees, where they're stocking food for their young and they're laying eggs and then sealing up the nest. It's just one female at a time. And when the bees and wasps are done, they fly off and the young overwinter inside the nest and then come out. The wasps are not pollinators the way bees are. They may use some floral resources, but they're not stocking that for their young inside the nest. So little different ecology, but if you put this kind of block out, that's what you're going to get. You're going to get both of them, and that's okay. They're really cool too. The kind of things that wasps stock are natural enemies, the wasps are natural enemies. The food that they stock for their babies is like caterpillars or aphids or things like that you'd maybe want to control in an area. So after they had hung their block and learned the different plugs, we asked them to check it like every 3 to 4 weeks, thinking that would be sort of a reasonable time commitment over the summer, and also provide us some information about when holes were plugged, when things were active. And then we had a nest block data entry webpage that we had built for us. And they would click on the holes and enter, if it was full or not on that particular observation day. And then, of course, the final thing that I don't have up here is they needed to return the blocks to us in the fall. And we actually reared them out to identify the young. So there's get to the staff work. So we had initial work of designing the project. And here's, again, this is the very similar to the Colorado Bees Needs project in the block design. There was a lot of work recruiting volunteers, getting the word out there, coordinating with volunteers, getting blocks to volunteers, getting blocks back from volunteers. That was pretty much Britt that did that. And then at the end of the season, the blocks come in, and that's when a lot of my work started. Actually ran from like October through April was my heaviest work period, bringing the blocks in, checking them in, seeing how many holes were there. We glued test tubes over the top of the holes so that when the bees came out, we knew, they were captured right there and we knew what hole they came from. They need to be overwintered in a cold storage. So there's nothing you can do about it. You have to go through a winter period before you get your adult bees with this method, which is fine. It just means you need to know that up front when you're doing your timelines. We used a walk-in growth chamber for rearing our bees, so we could put it down to about 5°C in the winter and then warmed them up. We warmed them a little early, starting in March, and warmed it up and checked them every day to catch bees in the tubes, which were then pinned and identified. And I'm ready, Britt. There we go. So some of the outcomes that came out of this, we got distributions. We got blocks all over the state, and we've got bees from some areas and not from other areas for some species. This map here is Minnesota. The different colors are biomes. The green is the coniferous forest area. The sort of reddish pink is the deciduous forest, the yellow is the grasslands, and then the purple up in the corner is tallgrass aspen. The blue, everything in blue is from our project, from the blocks. And the black sort of diamonds are what we had existing in the University of Minnesota insect collection. And so for this species, Megachile campanulae, we added a lot of new locations. And not only that, we know how many nests were in each location. So I can look at sort of relative population or relative abundance, at least in our blocks, in different areas. So the larger the blue circle, the more bees were in that area, or more nests were in our block from that area. Some more outcomes, we've got phenology data. Now these are some of our best phenology for 3 of our osmia, Lignaria, Pumila, and Tersula. And so we can see the Lignaria came out first, Pumila seemed to nest after that, and then Tersula sort of has a longer nesting period. We also have web pages up for them. Let's see. Some of the challenges, occasionally blocks would fail, get a crack in it. We had a squirrel eat part of one of the blocks. When we're looking at phenology information, we get date ranges. We don't get the specific date the nest was completed. We get, oh, they checked one day and there was nothing, and then they checked 3 weeks later and there was a plug. So between that time, we know it was completed. Another problem is, well, challenge, is that to get adult bees, we have to rear the whole nest and we kill all the offspring from that. So as far as a long-term monitoring, I don't see that as a great thing to kill, you know, all the young from one female. Some things that take a while. So overwintering, I mentioned, is it's just a part of the method. It's just something that you have to go through. The blocks can take up a fair amount of space. That's maybe a quarter of the blocks in the picture up there in the plastic bags on the shelves. And then there's a, you know, pinning an ID, which is typical for bee work. That takes some time. On the volunteer side of things, we got more people where there are more people in Minnesota. So the red pins are blocks, and then the map with the green and yellow and orange is where there are more people in Minnesota. People would sometimes confuse the plug materials with each other, so that wasn't always reliable. They were pretty good at timing, but then there's always some people that would hang a block and maybe not check it or not enter their data. So we got a range of different responses, which I think is normal and to be expected. It's just something to know when if you're planning a project. Ready. Some of the benefits. So we got comparable data across the state of Minnesota. This is a really large scale, geographic scale to cover. And we couldn't have done this without volunteers out there to hang blocks in different places for us. And we use the same method in 3 different years. So we know, you know, relative to each other within the state, within the blocks, where different bees are. It's fairly cost effective to build blocks and to send them out. And we have species level ID. So we have the specimens, we can look under the microscope and get that species level ID for all of them. And we're filling knowledge gaps about who's nesting and where they're nesting and what time they're nesting. This is a sneak preview of some distribution maps that we're hopefully going to get out in a publication here soon for a bunch of the different species. From the volunteer perspective, it's got a pretty high coolness factor. People seem to really like this. It's really engaging for them. We got a lot of people returning year after year to host blocks. It's something you can get your kids doing. You know, this is the blocks aren't going anywhere. You can always know where to go look for your bees. And for our staff, it's since it's a lot of winter and early spring work, it sort of can mesh with some other work schedules, like a normal field season during the summer. Ready. What's next for this portion of the project? We just got funding for like a second phase for the tunnel nesting bees, stem nesting bees. We're going to try to put blocks out all over the state again with similar phenology data and volunteer commitment, but we're also going to try to make it so we can open the blocks and look at the materials the bees are nesting and identify that using like DNA analysis and mass spec for the resin material. it's gonna build off the original website for now. So that's what I've got down there. And this is, so that picture up there, you can see where the bees have cut circles in the leaves. That's the kind of thing we started looking at with that iNaturalist project that Britt talked about. We're going to keep that going too. And I guess at this point, I'm handing it off to Elaine. Are you there, Elaine?
I am. I just had to find my mouse to unmute myself. So yeah, thanks, Colleen.
Great.
And hello, everyone. My name is Elaine Evans. I am an extension educator and researcher, and I work at the Bee Lab at the University of Minnesota. So I'm a bit in a couple of different worlds, but that all relate to each other. So for my extension work, I do a lot of public engagement and monitoring for pollinators, as well as just education about pollinator conservation. And then for my research work, I also work on pollinator conservation, do a lot with bumblebees. And so I have those kind of dual perspectives of being aware of engagement and education, but also thinking about how can these data be used. And that's something that was throughout the Bee Atlas project. We were really thinking about those, not just what volunteers can do, what the kind of data that they can get, but how can we fill in knowledge gaps? How can we have these data be something that will further the knowledge base. With the Bee Atlas project, I worked a lot on the bumblebee portion of things, and there's a lot of interest in bumblebees because they are one of the few groups within the bees where we have a good idea about how they're doing, and they're not doing great. So about between 1 out of 3 and 1 out of 4 species across North America, and the same pattern goes globally, are of conservation concern. Here in, you can go to the next one, Britt. Here in Minnesota, we have an endangered species here, so a federally listed endangered species, which happened after we had started the program, but we had an idea that this might be happening. But we are also concerned about a good number of other species in Minnesota and are really interested in making sure that we get information about them as potential listings come through. During the scope of the project, there was one other species, the yellow-banded bumblebee that we have in Minnesota that went through the process and was not listed. You can go to the next slide, Britt. So the bumblebees, using them, there's a lot of public interest in them, a lot of scientific interest in them, but there are some challenges to public participation with that conservation concern. And especially, once they were federally protected, we really needed to use non-lethal methods. When we were looking at building this project, we looked at models in other states, in Nebraska and in Vermont and in Maine, and a lot of projects at that point were based on collections. And fortunately, we had decided to go with non-lethal methods because once the federal listing was there, that's what we had to do. But even non-lethal methods, even just handling of bees, is discouraged in areas with documented endangered bee presence. And this is something that more people are going to have to think about as there are more potential listings. So there are other bees. Currently there aren't any federally listed bumblebees that are in the western part of the U.S., but there is potential for those listings. Oh wait, is Franklin and I actually listed now? I may be misspeaking. But anyway, there are these challenges with programs engaging the public when you're working with endangered species. If you can go to the to the next slide. There are things that make bumblebees more obvious choice for public participation, especially with the non-lethal methods. And one of that is that the species level ID for at least a subset of species, and it kind of depends on where you are regionally, but in Minnesota, we're lucky that a lot of our common species that we have are, it's teachable. With 3 or 4 hour workshop, people can learn how to tell apart some of the common species that are will make up 80% of the 80 to 90% of the bees that they're going to see out there. They're also, due to their size, they're more easily photographable. Next. So I mentioned that we were modeling our setup after some of these more collection-based surveys. One of the things that we wanted to do was to have equal survey effort across these different collection points. With iNaturalist, that was great for finding new places where species were, but we really just get occurrences and aren't able to say much other than that's where that bee is. And being interested in the population level of what's going on with bumblebees, we really wanted to be able to start to gather those data. So these volunteers were trained to ID those kind of simpler species that could be told apart from one another, supplemented with taking photos of all the other bees, all the other bumblebees. Next. For taking the photos, we did some training on that. We a lot of times in terms of hands-on instruction, we seemed to have bad luck with weather, where we tried to do a lot of the trainings in early spring or later spring, when bumblebees would be out, but before people needed to be really ready for surveys. But a lot of times we ended up with colder springs, and in our in-person events, it was hard to find bumblebees, but we did our best to try to show people and provide instruction for how to take photos. In terms of the ID information that we gave people, we tried to really be clear what photos we needed. And there's kind of simple things of get a photo of the front of the face, of the side of the body, of the top, so we can see the coloration on the abdomen, but also information about, here are 2 species that are really similar. We need you to this is the part on the face that we need to see. Please try to get a photo where we can see that part. And volunteers used different volunteers used different ways and kind of worked out their own ways for photographing. They're using, we were able to provide them with these photo tubes. So these photo tubes are based off of a design from James Thompson, who had, so there's a tube that's inside of the outer tube that has just a film, a camera lens, a clear camera lens on the bottom. And then the white part on the bottom there has a little divot. You can see the bee to the left of that photo tube is kind of in this little crevice. So we made a bee-sized crevice, and then you can have the bee in there and have them hold still. But it was still hard to get photos at all the different angles with them in there. So you could do that with bees that you just caught on flowers. But we did also have volunteers go out with coolers with ice to chill down the bees. And so then a lot of the photos were just from chilled bees. Some people also just took photos of bees as they were in containers. So they just had clear plastic containers that the bees were in. So these people were collecting bees from flowers. We had them blocked for 10 minutes at once, collecting all the bumblebees, taking photos, and the ones they didn't know, writing the ones they didn't know, and then releasing them. We used, in terms of where people were going, we used breeding bird routes. So these are routes that have been set up by the Breeding Bird Atlas Survey that are all across the U.S. and they are also set up to be spread out across different kind of ecological areas. This was something that worked well for us in Minnesota, where there's not a lot of public land, and where there is public land, it's really it's mostly just in the northern part of the state. If we were, to rely on people finding places, I was nervous about having people find places to go. So by using the Breeding Bird Atlas routes, we were able to set up these routes. Volunteers knew where to go, and these were all in publicly accessible roadsides. Let's see. Oh, good. the rest of the picture came in there. The routes we had, so the blue are just all of those routes that we had. So they weren't all claimed. We had a good amount of participation, and similar to what we saw with the STEM nests, is we had more participation where we had more people. We did have a good participation. We had routes that were near people's cabins up north. So the 5 here is the number of years. So we had some routes that were run for all 5 years of the project. Some people only ran them for 1 or 2 years. But we did have a lot of these routes were run consistently. And, we did a fair job of getting across to different parts of the state. Next. For outcomes, one of the things that we were able to do was to find new areas for occupancy with the rusty patch bumblebee. So the map there, those yellow blobs are areas where when the rusty patch bumblebee was listed in 2017, those were the areas that had documented rusty patch bumblebee were priority areas. They have the yellow spots kind of out from the one exact location to show where they're likely to be. So we had all these areas where we knew the patch bumblebee were likely to be, and we found 3 new spots. We were able to kind of expand those areas to show that there were rusty patch bumblebee present there. We also were able to learn some things about the bumblebee communities. We were looking at land use surrounding the bumblebees, surrounding those survey locations, and found some associations of different bumblebee species with either forested areas. Some were associated with developed areas, like possibly we didn't have a ton of rusty patch bumblebees, so I'm nervous about making too many claims. But in terms of, even though we were surveying across the state, away from developed areas, we still found most of the rusty patch bumblebees closer to developed areas. We also looked at the differences in abundances of bumblebees across those ecological provinces. So Colleen had mentioned, looking at those different ecological provinces in Minnesota, where we have this broadleaf forest and this mixed broadleaf and coniferous forest, and then we have prairie areas. And we did find a lower abundance of bumblebees in prairie areas. And the caveat there is that this was in roadsides in prairie areas. So, if our surveys were in other areas, we may have found them more. So there, maybe just differences in what those roadsides are like or what the surrounding habitat was like. Things that worked really well for this way that we did these bumblebee surveys. We had great volunteer recruitment and retention. We didn't get all of those routes covered, but we were, I was thinking we would be, maybe get 30 of them covered, and we got 45 of them covered. So I was happy in terms of how many routes we were able to have run. We had great retention with, I know the first year we had 100% retention with volunteers coming back the next year. There's some variability over the years, but we had people also then getting into it and adopting more than one route. We were able to have that equal effort across those surveys. So we are able to make these comparisons. I'm excited about this data set being a baseline information on bumblebee abundance, and we'll be able to use similar methods at another point in time in the future and really be able to say what's going on. A lot of the information we have about bumblebees is based on relative abundance. So we know how some species of bumblebees are doing in relation to other species of bumblebee, but it's always a question of what's happening with the actual abundance. I am happy with the geographic spread that we got. There are some parts of the state that we didn't have volunteers, especially up in the northwest corner of Minnesota. There are just not many people that are there. It's not an area of the state where people have cabins. It was difficult to get people there. I'd still love to know more about that. And actually, even in the southwest corner of the state, we had an interesting problem where people would do surveys there for 1 year, but then that's where the abundances of the bumblebees were lower, and a lot of times they would have routes where they didn't find any bumblebees, and we'd try to tell them, this is great information, we want you to keep going out there, but it's hard to keep volunteers engaged when they're just spending hours documenting no bumblebees. It's depressing, and it's not very fun, even though it's important data. You can go to the next one. So the ID quality, even though we tried our best to get reliable IDs, the ID quality was variable, and with that model of them, not taking photos of everything, some volunteers were incredible, and I trust their IDs more than my own at times, but some of them were questionable. So those volunteer reported IDs were not always reliable. Being limited to roadsides, we really also have limited conclusions we can make about habitat associations. Next slide. One of the problems with ID was species that are really similar. So we have two really similar species, vagans and sandersoni, and looking just at them from the top, they look the same. We really need to see these photos of the face. And even then, it's a pretty small difference where you can see the space below the eye and between the mandible is a little bit longer on vagans. That's really what we need to see on the females. So next slide. These are examples of a couple different kinds of photos we could get. That one on the left, that actually works. I can see the face, I can see that space, I can know that that is Bombus vagans. The one on the right, I have no idea. So we did ask for multiple photos, but bees are mobile, they're moving around. Not everybody consistently gave us enough reliable photos, so we ended up having to lump those two species. Next slide. So this is something I'm thinking about more lately with the potential listing as an endangered species of Bombus pennsylvanicus, which is the American bumblebee, which is currently being considered for listing as an endangered species. And this one is also difficult to ID unless from photos, unless you have really good photos of a few features for females, looking at the top of the head, and that orange line got a little messed up. But basically where the ocelli, where those little simple eyes are in relation to a line that you draw across the top of the big compound eyes, is one of the more reliable things, because unfortunately color patterns are variable in these two species and they overlap. Next slide. Another confusing thing here is it wouldn't just be lumping those two species, Pennsylvanicus and Oricomus, together, because the males are confusing for color patterns with two other species. So, and the males are even worse. There's some color pattern that you can rely on, but really the best way to know for sure is with specimens. And so potentially if you're lumping things together, you might have to lump together, 5 species, and that becomes less useful in terms of being able to really know what's going on. Next slide. So next steps, we do have a project where we're continuing to survey bumblebees. We've ended up over the last 5 years that we've been doing that, the Xerces Society has been expanding their state bumblebee atlases. And so we are now cooperating with them and using their methods, which we are using now a grid. So this was our first year. I was worried about volunteers finding publicly accessible lands, but they did. So this is what we're doing now. It seems to be working really well. Next slide. So I just have some conclusions overall for the Bee Atlas project, the Minnesota Bee Atlas project. iNaturalist was great in terms of low input from staff, broad engagement, and we got really important location data. For the stem nesting side of things, there was a lot of commitment from both volunteers and with staff time, but it did provide us with really important phenology and location data. For the bumblebees, it was lots of volunteer time. Staff time wasn't so much, it was mostly kind of photos and data management. But we're able to get abundance data, and some species were really tractable. I have one more slide here, and then we're done. I know I have a couple minutes over our slot. I did want to just mention, if you can go to the next slide, that
there is no next slide.
Oh, okay. Well, I was just going to mention the Biodiversity Atlas. So all of our records went into a Biodiversity Atlas, which is hosted by the Bell Museum, which is a natural history museum here at the University of Minnesota. And a nice thing there is we were able to include not just specimen data, which a lot of public databases are based on, but we were able to include those observational data. So if you go to the Bell Museum and find the Minnesota Biodiversity Atlas, all of our stem nesting and bumblebee records are included there, as well as bee records that we have from the University of Minnesota insect collection.
All right, thanks so much. That was such a great series of talks on a diversity of different ways that exploring distributions of bees across Minnesota. So thanks so much, Britt, Colleen, and Elaine. So right now, I'm going to hand it over to our next speaker, who's going to talk about a totally different type of project. So Andoni Melithopoulos, who's from the Oregon State University, is going to start out telling us about the Oregon Bee Atlas. So I'll let you take it away, Andoni.
Thanks so much. And you have two very similar looking people here. This is myself and Lincoln Best will be co-presenting. I'm going to kick things off, and but let me turn our camera off so that let me share a screen first. Oh, I see how this works. All right. Now I can't see nothing. Share. I'm going to do this. Share that like that. Okay. And I am, hopefully you can see the screen, and I'm going to turn this into, there, I see it now. Okay, great. Do that. And I'm going to change. Hopefully I'm not in presenter view anymore. Adrian, can you just confirm that?
That looks good. You can turn your camera off if you want the slides to be bigger.
Great. Let's just do this. That's right.
Assuming that your and Lincoln's voices are different enough, we can differentiate. We don't have to measure any Mallar spaces, I don't think. We might have lost your audio. Maybe you muted your audio instead of your camera.
I did, in fact.
Now I can hear you.
All right. There we go. Share screen. Share. Turn the camera off.
I think we're getting there.
Yay!
Now just switch. Perfect. Now we got it.
All right. Thank you so much.
Take it away.
Link and I are so honored to be invited to the summit to share our experiences with the Oregon Bee Atlas, which is a volunteer-led initiative to inventory the state's native bee fauna. And our co-authors include the various staff members of the Atlas, which are situated across 3 different departments at Oregon State University. And all of us serve a large group of rambunctious volunteer master melitologists who ultimately direct our activities. Oregon's path to a native bee monitoring program began with a tragedy, one that occurred on National Pollinator Week in 2013, when a pesticide applicator treated a highly attractive tree, a shade tree in a Target parking lot outside of Portland, resulting in thousands, 50,000 dead bees on the pavement. This incident garnered public concern and prompted the Oregon legislature to initiate a cross-agency collaboration known as the Oregon Bee Project. This statewide framework seems to have many parallels to the work that you're currently doing in Colorado. So let me just briefly mention it. The Oregon Bee Project is an extension-focused initiative centered around training and engaging a broad set of stakeholders involved in bee health or having some kind of touch point to bee health, providing them with decision-making support tools tailored to their needs or specific challenges, finding ways to showcase the innovators who are blazing the trail for bee health in their sector, and finally, developing a comprehensive way to measure the health of Oregon's 600 or so wild bees, the Oregon Bee Atlas. Now, the Atlas was meant to be a way to see if our extension and outreach work was ultimately having a real impact on the bees of Oregon. Now, as Dr. Carper and Messenger Carroll eloquently pointed out, serving for native bees is no mean feat. I do also want to make a shout out to Minnesota. It was their example, in fact, that gave us the push to get up and started in Oregon. So we are, in a sense, one of their grandchildren. And if it's all started in Colorado, I guess we're great-grandchildren. Now, this challenge, as Olivia pointed out, is not insurmountable. But the key challenge is succinctly put by John Asher on our weekly podcast, that surveying may need to be situated within a more basic task of inventorying bees and understanding their relationships to habitat, range maps, and such as, which is a point that Adrian raised in his introduction. Without this work being completed, we threaten to have a survey for generic and common bee taxa, which glosses over the rich diversity of bees we have in states like Oregon and Colorado. When it came to inventorying bees and habitats, we already had a model in Oregon to follow, namely the Flora of Oregon project, which was initiated in 1994, and it has managed to operate independently from a combination of grants and private donations to come up with this inventory. You can see it publishes these great books. They have a species inventory, they have range maps, and now they have a digital online interface operated through Symbiota. Being able, anybody who wants to do restoration work or have any look for the Flora of Oregon can track it down. Now, the sensibility of this approach is captured in the quote that starts the first volume of the Flora. Thomas Jefferson and Lewis Clark told Lewis and Clark to go out there and identify everything. Well, it's been 200 years, and it's high time we had an inventory. In a way, it's fitting that this should be such a grassroots sort of thing, the flora. Instead of a huge agency that could have put a few $1,000,000 into a flora project, but hasn't and won't, it's heartening to see that many dedicated people will get the job done. To this end, we knew to get this job done, we needed something akin to the native plant societies, filled with curious and independent melitologists. While the Flora was an example of a volunteer initiative that ended up meeting its goals, we were also conscious that many volunteer biodiversity initiatives succumb to certain pitfalls that we were conscious of when setting up the Atlas. The first pitfall is that volunteer capacities are frequently underestimated, resulting in relatively low investment in volunteer training and their narrow focus on some very basic tasks such as surveying for bees in something like this local park. The project stalls under the weight of thousands of new bee records of low scientific importance that have been poorly prepared and with location information that has either not been databased or is riddled with errors. This results in a bottleneck where taxonomic experts who can identify the bees get overwhelmed and their productivity increasingly diminishes. The taxonomic bottleneck in turn has a corrosive effect on the volunteers. If volunteers are lucky, years will pass before they get information back on their discoveries. More often than not, the discoveries disappear down a black hole, either because the material never gets determined or identified, or if it does, it gets whisked into a scientific publication without a thread connecting it back to their effort. Volunteers get discouraged and fall away. So to get around these challenges, we decided to emphasize what we do well in Extension, investment in developing our volunteers. And we knew this investment would pay off because surveys performed by our team evaluator Heather Fisher with the OSU STEM Research Center showed that fully 43% of volunteers had very high ambitions for exploration and discovery. And I do think this is connected to a phenomena that can be traced back to Joe Wilson and Olivia's book, Bees in Your Backyard, where suddenly there was a whole crop of people who are hungry to dig a lot deeper into native bee biology. Evidence of this large number of dedicated and well-educated volunteers who are willing to put in the time to do a lot more than most projects expected became quickly apparent to us. A great example of this can be seen in the collection submitted a few winters ago by master melitologist Ellen Waterous, a retired professional with no previous experience in entomology. Not only are specimens prepared to museum quality, but she took the time to work through 24 monographs on the family Adrena and successfully identified most of these bees to the subgenus level. For these and many feats, we love puns, she was awarded our annual Determinator Award. Successes like Ellen's are the product of an intensive investment in volunteer education through a multi-year program called the Master Melitologist Program, which is overseen by Jen Larson and Sarah Kincaid. For those of you who are in cooperative extension, you know that this is the kind of education and volunteer management that we excel at. All participants are required to complete a self-paced 6-module online training covering topics such as bee biology, database management, survey techniques, and specimen preparation. The training focuses on practical technique development and includes interactive activities such as this pinning assignment shown here, where the instructor can grade the participants' work using uploaded photographs of the volunteers' growing collection. Notably, this online platform has enabled us to deliver the education to volunteers outside the state, and we now have melitologists in training in Washington, Idaho, and British Columbia. Key to the training is making sure students understand how to capture records. Core to the Atlas is a workflow that does not encumber the volunteers in paperwork, but rather facilitates their own discovery of the state's native bee fauna in a manner that leads to highly accurate records. Here you can see volunteers Martha Richards, and in a second you're going to see Pam Arian, combing through a patch of vetch and camas in the mid-Columbia region. What they do is collect the plant record. They're collecting the plant record, not the bee record in iNaturalist. They then pool all the bees into a single killing jar from the location, and then they associate the bees in the jar with a unique sample ID for the day. Plant and location data is captured using iNaturalist. We then extract these records and reformat them using a Python script and reassemble them in an online database where volunteers can verify their location information. We then pull those records out to generate location labels those small little labels that go on the insects that we mail out to each volunteer twice per month. And volunteers identify this as one of their biggest fears, is how are they going to make those labels? At the end of the season, we have another tab where volunteers enter their determinations with a drop-down menu to ensure that there are no misspellings of the genus or common species names. Not only is this exceedingly efficient and error-free, but it's resulting in one of the largest databases of bee host plant records anywhere. We supplement our online training with in-person training where instructors demonstrate survey techniques and how to use genus and species level keys. This includes our 5-day native bee identification course that we hope to offer again in August next year. Combined, this has resulted in volunteers who are motivated, knowledgeable, and starting to make remarkable contributions to our knowledge of the bees of Oregon. Volunteers in return get information like this. Everybody gets a report once a year that helps them focus and redirect their effort over time. So this is one of our volunteers in Central Oregon. You can see it shows them the species that they collected, the location of those records, and provides a whole lot of other information about the phenology of the bees and how they're doing relative to everybody else in the program. And they get one of these back 12 months after submitting their collections. Volunteers submit their collection a month before our annual conference in March, which allows us to get feedback on the preparation of their specimens by the conference. And it also allows us to bring in native bee biologists of some repute to get everybody pumped up for the next year. Now, this has resulted in a large volume of fully database bees and volunteers like Heike, who have grown into budding and increasingly independent and curious melitologists. Here you can see our lab with a subset of the collections from volunteers collected after 2020. It's a big job to go through all these bees and confirm the identification provided by volunteers. And at this point, and at this stage in the whole process, let me introduce Lincoln Best. He is the person who does all of this work and goes through all those bees. And I will say, having a taxonomist on staff and paid for is a key element to having this kind of work done. So come on up, Link.
Okay. Hi, everyone. I'm Lincoln Best. I'm the taxonomist for the Oregon Bee Atlas. Oh, there we go. Got a laser pointer. Oh, and moving forward. So as you've seen, I am the bottleneck. We bring in thousands and thousands or tens of thousands of bee specimens in our annual cycle, and they all come to me. And I spend the winters on the microscope, identifying as many as I can to either species or the generic level. And then we upload those identifications to our data set. We identify vouchers for each species from each county for each collecting year, and those end up in the Oregon State Arthropod collection, which is managed by Chris Marshall. And then ultimately, those records will get published online. Through Oregon State Arthropod collection, we serve up our species occurrence data using the OSAC integrated publishing toolkit. And so then that data becomes publicly available on GBIF and other biodiversity data aggregators. Now, what is a bee atlas without an actual atlas? And so we're lucky to have Chris Marshall working on developing an online interactive Oregon Bee Atlas. And the goal of this atlas will be to showcase the immense amount of data we've produced, but also provide the public, researchers, and others with tools to better access that data. And so we want to make it possible for people to extract different kinds of data from our giant data set. So things you might consider would be just downloading the different bees that occur in a limited area. So if you are restoring a state park, you would be able to potentially download all of the bee records we have for your state park, or for your ecoregion, or for your county, or for some other area. Additionally, we'd like people to be able to download the bees associated with different plants. And so we all love to make plant lists. And so this might be a way to identify high priority plants in different regions for restoration projects or other reasons. Now, as Adoni mentioned, all of our samples, our data is being captured through iNaturalist. And I see that Minnesota has been using iNaturalist for similar reasons. And so, as Adoni mentioned, our investigators go around, take an image of a plant, upload it to iNat, and then that also serves as the sample data for our bees. And you can see here that we have more than 14,000 unique samples. So collections of bees sampled off some particular plant at some particular place and time. And those bees have been sampled from more than 1,250 flowering plant species in the state by more than 180 people. You can kind of see our champions here. Some of you may know Pete and Gretchen Peterson. They worked in the Colorado backcountry for much of their lives, and they've collected nearly 10% of all of our samples. And you can see here as well that some of our master melittologists have collected bees from more than 290 flowering plant species. That's really impressive. It's a lot of plants and a lot of samples. So just taking a quick look at some of our sample data, as I mentioned, our sample runs in somewhat of an annual cycle. So we have, of course, some training in the spring, which is when we usually intake our master melittologist apprentices. And then we have a super fun field season where people travel all over the state, taking pictures of plants and sampling bees from them. And then we have a fall where individuals are working to identify their own bees, and then they submit their bees at the end of the winter, and then I take over from there. So in 2018, we found that our participants collected a few thousand samples. We can see that they're really quite dense around Bend, in the Willamette Valley, Portland, Corvallis, Salem area, and Eugene, as well as in the Rogue River in the Southwest. And that's not a surprise. Those are our major population centers in the state. And out here in the high desert, we find that there's far fewer records, as well as through the Blue Mountains here into the Wallowas in the northeast part of the state. But with some instruction and some guidance, in our second annual cycle, we found that people were able to get out into the high Cascades more, along the coast, sampling all those cool coastal dune habitats, and also getting into the high desert, the Blue Mountains, and out into the Wallowas, and all around the Umatillas as well. So that was really impressive. And another year later, even more effort in remote parts of the state has given us much more even sampling spatially. And finally, if we include, of course, all those 14,000 records on iNaturalist, we find that the state has quite even coverage. We still have a couple, what I describe as black holes spatially, in the east part of the high desert, and in the Umpqua. For some reason, we have very low sampling intensity in the Umpqua watershed. But otherwise, we have really, really good sampling. And we have some individuals that are going on special missions to run trap lines and do biweekly sampling in areas like Lakeview County that there's just nothing out there except sagebrush and wildflowers and elk. And that's a lot of samples, 14,000 plus samples. We find that each sample represents about 6 or 7 bees on average. And so that's nearly 100,000 specimens. And of course, if we include other sampling methods that our project is using, like vein traps, it's far in excess of 100,000 specimens. Now, in a Bee Atlas talk, we need to talk about two things that we all love. One is flowers. And so when you enter the Master Melittologist program, you may not be an expert on wildflowers or botany or plant identification, and that's fine because we provide you with a simple hit list to get you started. And these are all quite well-known plants. You can see through the phenology that we provide options all across the flowering season. And for those of you that don't know all these Latin names, I can tell you that we have thistles and rabbit brush and gumweed and sunflowers and goldenrod, wire lettuce and phacelia and popcorn flowers and cacti. So these are common plants that most people are familiar with. They're really important parts of their plant communities wherever they occur, and they attract lots of bees, so a great abundance of bees and other pollinators. But they also attract a lot of weird bees as well. So things like all of these asters, they get many, many specialist bees that only want to visit wire lettuce in the desert or goldenrod in the fall. Gives you lots and lots of things that you can find. And also not everyone has the ability to travel to, you know, the alpine out in the high desert. And so this gives people the opportunity to find cool bees and do some really good documentation local to where they are. You can find some of these certainly on your street or in a nearby park. And so it gives everyone the option to contribute. Now, we're really lucky in Oregon, as Adoni mentioned earlier, to have the Oregon Flora Project. And their new website is really amazing. If you haven't seen it, I encourage you to go over and check out the Oregon Flora Project website. It's amazing. So we use tools like this to help inform and educate our bee hunters in conjunction with that plant hit list, so that they can go out and they can find some of these interesting, in this case, Plagiobothris popcorn flowers. Now, not only do we have the hit list, but sometimes we want to go out and find really obscure specific things. So instead of sending people out to survey on goldenrod, we'll send them, in this case for 2021, we sent them out to find Techelia natalii, which is a tiny little desert plant with an even smaller flower, which is covered in these spiny little trichomes on their leaves. It's very nondescript and it's very small, and it occurs in a really remote part of the state. It's probably widespread, but there isn't a ton of data for Oregon. And the genus and species is much more abundant from the further southwest you get into the other desert systems. And so if you go on the Oregon Flora, you can get this treasure map of where the Techelia is found in Oregon. We have a patch over by Pendleton, and then we have it sporadically through the high desert here. And so we did have some master melitologists go out there and find a giant patch of Techelia out south of the Alvord Desert. And sure enough, what did they find? Well, they found this rare little fairy bee that only collects pollen from that genus of plants. And so thanks to Zach Portman's 2016 treatment of this interesting subgenus, we now are able to go out and document these little checker-spotted fairy bees, which otherwise we would not likely be able to find very easily. Now, among those 1,250 species of plants that we've documented, well, it's a lot. And so one way to visualize that is to just take one of the 400 genera that has been sampled on, and then to take a look at the species within that genus. And so here I'm just providing an example of penstemons, because penstemons are fairly showy and beautiful and widespread in various ecosystems. And what we found was our master melitologists have collected bees from more than 30 species of penstemons, from the desert to the alpine to moist meadows and everywhere in between. And some of those penstemons are actually rare. And so penstemon peckii and penstemon hesperius, which we have a handful of bee samples from, are quite rare, likely in need of protection. And so we're able to add information about the pollinators for those rare species, which is really value added. And so our master melitologists have collected more than 289 bee samples from penstemons, likely representing more than 2,000 bee specimens. Our penstemons have been, records on iNaturalist have been curated by more than 26 identifiers, and all of these samples were collected by 48 of our master melitologists, which is pretty awesome. I mean, penstemons are amazing. Now, what's a bee talk without a whole bunch of bees? So among those 100,000 bee records, we'll take a look at one genus, and that is Anthophora. These are the digger bees. Anthophora occur all over the state in almost all of our habitats. There's spring species and summer species and late summer species. There's generalist foragers and more specialized foragers. And here you can see that we have sampling of the genus almost everywhere that we've had sampling. And so let's take a look at some of these species. We find that Anthophora urbana, pictured on the right, is widespread, abundant, and really common in the state. And so is Anthophora bomboides, fairly widespread, fairly common. One trend you'll see in any kind of sampling program is that very common things are documented very often, and very rare things are documented very rarely. And so what we find is we have some species of Anthophora that we find all over the place and in great numbers. And then we find all of these other species in very few numbers. So we have here Anthophora porterae found in just 2 locations in the States so far. And Anthophora ursina with these great face tattoos on the male is found fairly widespread, but not in the Northwest, where it's much moister. Now, a handful of our species, you'll see, are quite range limited and very uncommon. So we're pretty confident at this point that Anthophora californica is only going to be found in the southwest part of the state in the Siskiyou and Klamath regions. And Anthophora edwardsii, known from only a single record, has been found in the Wallowas. And this little bee in the male has a tiny little spur on its back leg. So those are the things I'm looking for all winter long through tens and tens of thousands of specimens. We find that Anthophora affabilis is also known from one specimen in the state down in the high desert near Lakeview. So things that are very rare, difficult to detect, maybe out for a short period of time or only really associated with a few plant species become increasingly difficult to find. Now, Anthophora albata might be my favorite Oregon bee, at least right now, and it's been found 3 times from Bend to Brothers, and the females have these giant eyes, and they're all white, whereas the males have all these crazy secondary sexual characteristics. They have these black flags on their wrists, and their hind legs are modified into these really grotesque shapes. And we have so many bees like this. Isolated, we find that Anthophora curta is only known from on the Columbia River north of the Umatillas right here. We find that Anthophora exigua is found in the high desert from Burns down to the Steens and in the Alvord Desert. We find that Anthophora lesquerelle is known from one specimen where the bootleg fire went through the Fremont-Winema National Forest this past summer. I couldn't find any photos, so this is another one of our Sasquatch bees. And Anthophora maculifrons, which is a bee with pretty crazy face tattoos, is found from Bend down again into the high desert, but rather infrequently, given how much sampling we have going on. Finally, we have a single record for Anthophora peritome, which was found at Tony's Ranch just one time. Now, we only have one record of this bee, but this bee isn't really that rare. It's kind of widespread in the West, and it can be locally abundant. But even with the amount of sampling that we have going on, a lot of these things still are difficult to find. But once you've found them once, now we know where there's populations and some of their floral relations, and we can go back and we can repeat those observations. Now, this is a fair amount just for the genus of digger bees, Anthophora. For the family Megachelidae, we've found nearly 200 species in the last couple of years, and we figure that there may be as many as 50 more species to add to this list. So we find that Coleoxys are the cuckoos of leafcutter bees, Dianthidium are resin bees, as are Hyriades. Hoplites have some really cool floral associations. The Megachile are our true leafcutter bees, but of course also use masonry and resins to make their nests. Osmia are our mason bees. We estimate there's more than 75 species of mason bees in the state of Oregon, which is super cool. They're in every ecoregion. They have all sorts of cool floral associations, which I love. So we've gone through about 4 annual cycles now. We have generated probably in excess of 100,000 bee specimens with bee-plant relationship data collected. These are for more than 500 bee species, collected from more than 1,250 different flowering plant species. And of course, these are widely distributed across the state and across the flowering season. We hope to have about 51,000 records published on GBIF by the end of the year, so please stay tuned. And if you're interested in becoming a master melittologist or learning more about the program, I put the link in the chat, and you can head over to the OregonBeeAtlas.org website to check that out. Now, we certainly can't do this alone. We're fortunate to have around 200 master melittologists doing a lot of this work for us, and we've been really lucky to find grant support through FFAR, the USDA NIFA, ARF grants, and of course, we're very fortunate to have support from Jerry and Judith, Ray Temple, and Stephanie Hazen, Missy Martin, and Jean Natter. Thank you.
Awesome. Thanks so much. What a great informative talk. At this point in time, we've got a handful of questions in the question and answer, but I'm going to actually invite some other guests to join me up here if they want, in case they have questions, or if in the discussion, if all of our presenters want to turn their cameras back on, if they're still here, we'll take a few question and answers and have a little bit of discussion. So just be careful, mindful of any cables there. We can pass microphones back and forth. So, can you stop your screen sharing, Andoni?
Oh, yeah, sorry.
How do I do that?
Oh, we thought we did.
I think, did they get it?
Well, maybe we did and it's back.
Yeah, I think we got it now.
Okay, great.
Perfect. I mean, what a great series of talks. You know, I'm biased as a researcher myself. I'm sure you guys are as well. So let me give a quick introduction for you guys and for people watching. You've met Olivia already from Virginia. Scott down on the end is our resident taxonomic expert in bees, and Lisa Mason here is at CSU. She's been running a community science project for some years in Colorado. So they probably have questions or can help me ask you guys questions about your projects. So I don't know, anything burning? I've got lots of notes here.
I have a burning.
Oh, they have burning questions. I'll let Olivia go first.
Oh.
Okay. Well, Virginia, you have a lot.
Okay, I just wondered, in addition to gathering photographs and getting additional plant records that way for your collecting sites, have you worked with any herbaria, or have you talked with anyone about actually bringing in herbarium specimens in addition to bee specimens?
I'm guessing this is for
Oregon.
Andoni.
Oh, for Oregon.
Yeah.
We have considered it. And we, you see that our volunteers are extremely talented, but we don't have the capacity. And any additional thing we take on is a problem that needs to be solved. We would love to be able to get involved in more plant taxonomy because there's so much going on. We just don't have the capacity. There's just a couple of us here, and it would either be put on us or it would be put on the herbarium and the Oregon Flora, and they don't have capacity either for the most part. But that said, a lot of our members who are very skilled botanists, they already take plant specimens in order to elucidate the identities of their plants in the iNaturalist record. So some of them are supported in that way. It's just not vouchered through our herbarium on campus.
Okay, I totally understand that issue with becoming overwhelmed with projects.
And I just want to say, like, what an impressive project both of these are. I mean, Oregon in particular, to be only a couple of folks organizing statewide inventorying that's specimen collection based is super impressive. So maybe I'll just go ahead and ask my biggest question, and it's really from both of your perspectives, which is really about the trade-off between specimen-based collection and using newer tools like iNaturalist. So I know that it's really impressive how you've integrated the two for the Oregon project, but it's still primarily specimen collection based. And so I'm curious to see what the Minnesota folks think of that, or what limitations using iNaturalist primarily are.
For us in Minnesota, I mean, it's pretty clear cut. We can't have volunteers going out and doing lethal collection unless they have a permit from the U.S. Fish and Wildlife Service. So that's kind of burdensome to try to get, permitted, hundreds of people on permits and have them trained and all that kind of stuff. So, we just are, because of, the obligation for protecting endangered species, we can't have volunteers do lethal collection. At least for anything where they might be getting bumblebees. Stem nesting, like Colleen was talking about, they'd like to reduce that, but we know bumblebees aren't going to go into the trap nest. But if we just have people out, collecting any bees off of flowers, you can tell them to not get bumblebees, but it's still a risk.
And so for Andoni and Lincoln, have you given consideration to how your design will be forced to change with the new listing of Franklin's bumblebee?
We've given it some thought. We work with the U.S. Fish and Wildlife Service locally. With respect to our lethal sampling program and the netting program, a lot of our members just don't collect bumblebees already.
Well, it's in our Forest Service permits.
And also it's already in our Forest Service permits to avoid bumblebees. And so our training is really good, and our members are super capable. And so they can recognize bumblebees and avoid bumblebees in the lethal netting sampling regime. Of course, trapping, that'll have an impact on trapping. And there's a lot of trade-offs here, because lethal sampling can generate magnitudes more data than photo documentation can. And if we're talking about always being data depauperate, we're not going to be data informed through photo documentation. And so there's a real trade-off, and we're going to have to reconcile that somehow.
Other questions from our panel?
I just have to commend all of you. There's 5, right? 5 total. Your projects are so incredible and so informative, and I look forward to the day that something similar is being done in New Mexico. I was thinking, Colleen, about I think it was Colleen, you were the one that was talking about the bee blocks. Did you drill all of the holes for those? How does...
Actually, we were lucky, and we had some volunteers that were able to help with that. So Rick looked around and coordinated and found some. Actually, it wasn't all volunteer. There was one volunteer that did quite a lot of them for us, and then there was some that we contracted out to have made for us. But in the next phase of the project, we're looking at a new design where we can open the traps up more easily. Yeah, that's a lot of drilling. So far it's just me and my staff on that, but we might be looking for volunteers with woodworking skills, and they're out there. It's just kind of, finding and keeping people around. So.
That makes sense. Both of your projects, I think, really highlight how much volunteers can do to help us move forward with understanding bees. Yeah, it's just really cool to see. Link, I was wondering, do you guys record zeros through iNaturalist? Is there a way, if you have a plant and you don't get any bees on it, to just record that you looked and nothing was there?
So we've faced multiple revolts from volunteers, coming back to the old spaghetti factory revolt of 2018. And we've tried that. And so we had a field, because I really, I talked to Jim Kane. He said, here's a protocol. And they just don't like it. A lot of them are retired professionals, and they don't want they want to find a lot of bees, crazy bees, and they don't want paperwork. So we've got a problem there. Sorry, go ahead, Mike.
Yeah, I don't have anything else to add, but we had to take the old spaghetti house revolt very seriously. So unfortunately, we are not collecting zeros at this time for plants that are sampled that there's no bees visited.
Similarly, with the Minnesota, the trap nesting, did you have bee blocks that had nothing in them?
Yes. Usually there was something like a spider or something in there, or maybe some wasps, but there were some that had less and some that had more. So in that case, we do have a out of 30 count for every block, pretty much, whether there were bees or not. So that lets me do a little bit more of like a comparable abundance between one area or another.
We're at noon, but I'd like to keep asking a couple of questions, if that's okay with you guys. So for our attendees, feel free to stay here and listen on to some questions. If you need to take a pit stop, for all means, run for it. But I think, one of the most important things in terms of engaging with your community of mellotologists and bee block runners and everything is having positive feedback for them. So, it's impressive what the Oregon Bee Atlas does in terms of feedback. Like for Minnesota, when you're using iNaturalist, you get that immediate feedback of like, oh, John Asher just identified my bee. Right? Do y'all guys do any other kind of positive like reinforcement to engage with your community scientists?
I can take that one as the person who had the most volunteer contact. And it depends on, what I had available to contact them, or, which way. So if they had formally signed up to host a bee block, I could look in, I could see how they contributed data, have they not, do they need a reminder? Can I send a thanks for always being prompt message? For iNaturalist, it got to be that anybody can contribute from anywhere. And so they may or may not have known about our project or been affiliated. And so I really relied on iNaturalist and the systems in there to work with them. Similarly, with bumblebees, I would say same as bee blocks, in that they had signed up, they had been dedicated. So we could look at what data was coming in. And then there was always an end of season wrap up. So here is a final picture of your bee block. Here's the list of species that we found, how many of each one, what hole did they come out of? And here's what the statewide data looked like. So how did yours compare to that? Elaine and Colleen, anything else you want to add?
Yeah, well, just one of the kind of feedback things that was difficult is, when people found Rusty Patch Bumblebee, they were super excited about it, and they knew immediately how important their data were, because we told them, as soon as you find one, contact us, and we would share it with the Fish and Wildlife Service. But then we'd have to say, okay, now you can't go there anymore, and we'd have to change their route. And so that was, I know that was disappointing for those volunteers, but I mean, they knew that they contributed something really important, but then then we had to shift things.
And kind of along the same lines that Adrian asked, so after the volunteers take this training, after they pass the master methodologist course, I think you said that correctly, how do you continue to engage them so that you're getting good data, but the volunteers also have a really great experience in the program?
Well, I'll start. I just take ideas from everybody, including Minnesota. I have to say, early on, the kind of volunteer feedback material that they provided, I copied incessantly. There were no royalties, checks in the mail. But I would say that what we've developed, we looked at other master certificate programs, and they all have levels. So people come in at the journey level, being able to collect and database bees. That's the competency of a journey level. The apprentice level is going to be mastering the genus level key. So they're going to be given a box of bees, and so they got a score. They've got a month or so to work through them, and they have to identify a certain percentage correctly. We're contemplating the master's level, but this is where it gets stuck, because I've seen this in master beekeeping program kind of become a quagmire. we want people to do independent projects. And trying to figure out how to do that doesn't swamp us in, supervising 6 people is something that we need help with, and we're trying to figure out. There's one way we could do it where we give you a species level test, but clearly at that point we would like to get beyond just identification and making kind of significant. I think, Michael O'Loughlin and his brother Dan were looking for nomia, and they contacted soil scientists. They got data layers. They made these intricate maps. I didn't even know about this. And so clearly we have to find a way to encourage that and recognize it without kind of swamping us in projects. And so you identified a key aspect. People are going to need that to keep moving. But how to do it in a way that doesn't overwhelm us is going to be a trick.
Well, very well said. I could stand here and ask questions all day, but we're going to have to take a break for lunch. I really want to thank all of you guys for giving talks, sharing information about your projects with us. If y'all can hang around for the rest of the summit, I'm sure there are people that would love to chat you guys up. So I'll be looking for you there. And I'll just take this opportunity to remind folks that we're going to have a happy hour. So I'm going to be there. Hopefully you guys are all going to be there. So if you've got other questions, or if you guys want to hang out with us, find us on there at from 3:30 to 4:30. So we're going to take a little lunch break now for everybody, and take this opportunity to, again, check out our exhibitors or our sponsors, and I'll see you back here at 12:30. Thanks so much.
Bye.
All right.
How does the coffee work? Oh, let's see.
That's it.